especially Harish and Lakshmi, who were calling me. And I'm really thankful to the Vice Chancellor and DR for recognizing the little work done, my tenure at IHR. Actually, for past 3 decades, I was more concentrating on biotic stress resistance. As Harish told, Madam, there are many talks, you diversify your topic. So I've chosen this topic. We are just beginners here. I will be presenting briefly about the work done at IHR as well as at other places. I'll straight go to my topic. As we all know, Chilli got introduced to our country 533 years back due to its cross-pollination. Almost 90% cross-pollination has been reported. Depending upon the temperature, the stigma exertion will be there. So naturally cross-pollination occurred, and based on our own farmer selections, a lot of land assets have been evolved in our country. One of the best examples is Badigi chili, what we were discussing from the morning for its color. And in northeast region, that Bhut Jolokia for pungency. These are the major bioactive compounds of capsicum species, mainly capsaicinoids and carotenoids. These are the major biotic compounds. Altogether, the oil extracted is oleoresin, as we all know. And there is alternatively, for capsaicinoids, there is another compound called capsinoids, where people cannot eat the pungent chili, but with all medicinal values of capsaicinoids, there is a scope for exploiting capsinoids in capsicum species, which are very specific to capsicum genus. As you all know, apart from this, it's really very rich in vitamin C. I got wondered when Dr. Pal, our physiologist in our institute, when he has estimated vitamin C content in all my germplasm, it is having richest vitamin C content as a green fruit. As it is water soluble, if we eat raw only, we will get it. It's more than citrus fruit. It's very rich in vitamin C, which are having nutraceutical value of anti-cancerous, and vitamin E, vitamin K, and chlorophyll and carotenoids, lutein. This is also a very important compound, and beta-carotene and zeaxanthin. Zeaxanthin and lutein are being used exclusively in eye drops, and some companies are extracting and exporting to other countries. And zeaxanthin, cryptoxanthin, capsanthin, capsirubin, and anthocyanins. These are the major compounds which are very important for having nutraceutical property, and capsicum is very rich, and there is a lot of scope to improve all these compounds in our varieties which we are going to release. And the byproducts, you can see what are available in the market. Actually, compared to India, recently I was in US, there are plenty of products like in the market in US compared to India. Even the top chocolates, ice cream, everything chili flavor, and they give a lot of importance. There is a huge scope. The foreign exchange, what we are earning, around 12,500 crores we are earning by exporting chili. In that, oleoresin oil itself has a very huge share in that. So there is a huge scope. As the demand is more in our country, we are almost consuming 95% of the produce what we are producing in India. Only 5% we are exporting. With that, we are earning almost 12,500 crores every year. So coming to the industrial use, since morning we were talking about how important it is in food industry, pharmaceutical industry, cosmetic industry, and the pesticide industry. Actually, it is also useful in seed industry, which I am not highlighting here. These are the 4 industries which mainly it is used. The bio products are being used. It is there for the health benefits are known since beginning in India, even in Ayurveda, in our traditional use. Earlier it was used as for toothache, for gastroenteritis disorders, and diabetes, muscular pain, and weight loss also. swallowing like a tablet. It is traditionally being followed in our country, and a lot of health benefits have been proved, and a lot of work on anti-cancerous, prostate cancer, breast cancer, a lot of work is being conducted with the capsaicin. For the breeders, the mainly we have to look, of course, we look into, always look into disease resistance and yield, and quality is also equally important. Of course, fruit weight and the fruit dry matter content, which is more important for the breeders. Next, pericarp thickness for quick drying. Apart from that, capsaicinoids, oleoresin and color. These are the main quality attributes which we have to look into, apart from disease resistance and yield. Without quality, our varieties, no farmer will accept. Capsaicinoids, as you see, it ranges from 0 to 1 million SHU. Now, people are breeding for high pungency in chinense, and now almost all 2.39 to 2.5 million SHU is also available right now. Capsaicinoids are amides produced in capsicum species. They consist 3 different forms. It is available. Capsaicinoids, 3 fractions: capsaicin, dihydrocapsaicin, and non-dihydrocapsaicin. 90% of the pungency is due to capsaicin and dihydrocapsaicin, but I think I have mentioned somewhere. Only around 9 to 10% is dihydrocapsaicin, which is very important for retaining the pungency for longer period, even during the storage. So while breeding, we may have to look into, because when market is not there, people normally store in the cold storages. The pungency need to be retained, means we have to look into dihydrocapsaicin content in the chili. As we know, it's a highly volatile with pungent. It's a colorless crystalline to waxy substance. It is due to activation of TRPV receptor, which is involved in pain pathways, and which has promoted much basic and clinical research on the use of capsaicinoids. As I was telling, the capsaicin, dihydrocapsaicin, 90% of the fractions of capsaicinoids is due to those two, and non-dihydrocapsaicin is having only 7% contributing towards pungency, which is more important while breeding for pungency. A synthesis, we know where it is, the vesicles of the placenta, which are called blisters. This is the place where the pungency is located. Looking into the anatomy of the chili fruit, the interlocular septa, where the blisters are there the pungency is concentrated. And a lot of work has been done across the world. Genetic resources have been identified for capsaicinoid compounds in capsicum, and I have just reviewed the papers here. Throughout the world, many people have published many papers where the levels of capsaicin, non-dihydrocapsaicin and dihydrocapsaicin content has been presented already. It is available. Now, in chinense group, people started working. Initially, it was Bujolokia, which entered into the Guinness Book of World Record because of its highest pungency. But later on, people started breeding for it, and now right now the hottest pepper is pepper X, which is bred by Ed Curie, which is having 2.69 million SHU. It has entered into Guinness Book in 2023. These are the pictures, hottest peppers in the world. But these are all chinense group. Our concentration should be as these peppers will be well grown under shade or at higher altitudes like northeastern region. But in the plains, if you want to grow, it's slightly difficult. The yield will be very low. So we have to breed, we have to transfer this pungency into annum, which can be cultivated in the plains, in tropical areas. That will be of more useful to the farmers. Genetics has been studied, as somebody has already told. It is initially it was mentioned a single dominant gene, but it is a quantitatively inherited one. And there are reports that it's a polygenic additive. That's Dr. Naresh, my student, only did for his MSc work, and we found it is a polygenic. And other authors also mentioned that it is due to major and minor genes. And the markers are associated with pungency are also published by many, mainly located on chromosome 2, 3, and 7. There are a lot many publications on markers. Oh God. And Han et al., they found through GWAS and QTL mapping, they found identified 5 QTLs located on 2, 3, 4, and 10. These are the 5 genes which were identified as candidates controlling capsaicinoid content. Coming to capsinoids, as I told, they are low pungent capsinoid analogs which were first isolated from the low pungent capsicum annum cultivar CH19 sweet. This was first reported from Japan. These were people who were having allergy towards capsaicin, the pungency which they cannot consume. This is a good alternative. It is having just similar structure of capsaicinoids, but have ester group instead of amide group. That is only the difference between these two. But it has same medicinal value as capsaicinoids have. So this is another good alternative for the breeders to look into. These are the genotypes which are having very good level of capsinoids which has been published. And we also worked at IIHR. We could identify top 5 capsicum accessions with highest capsinoids, and the accessions having both. And we have studied the stability. All biochemical compounds except NAR dihydrocapsaicin and dihydrocapsiate are less influenced by the environment. Other compounds are less influenced. These are the 2 compounds which are getting influenced by the environment. They are not that stable. Actually, so I got capsicum Bujolokia fruits from northeast. When we analyzed at IHR, it was having almost 1 million SHU. When same seed have grown at IHR, it has only, those fruits has only 50,000 SHU. That much was the reduction. So G into E interaction is very much in pungency especially. Even morning Gadagimat sir was discussing about the Badigi chili, why other institutes are not working. That has a GI tag of from this area of your main chili growing area, and lot of environment has, because volatile compounds, we also collected lot many collections across Karnataka, and we have grown at Bangalore, but that flavor is missing. So we thought that is not the right place to breed for, that's what I wanted to tell sir. This region people only Dharwad, Devi Hosur, these area people, if you work, you will get better Badigi types if you want. Otherwise, we can breed for Badigi-like chilies, Badigi-like hybrids. And when we have done the stability analysis, the genotypes which are more stable for capsaicinoids and capsinoids across the seasons I have mentioned here. This is one of my PhD students' work. Next slide, please. Can you change the slide? Yeah, we did genetic studies we have done. We are given the gene effects of dominance and additive into additive type of gene action, predominant for capsaicin, dihydrocapsaicin, and total capsaicinoids, capsiate and dihydrocapsiate and total capsiate. This was the genetics we have studied, whereas for NAR dihydrocapsaicin, the genetics is additive and additive into additive type of interaction we found. Oh God. And we did little marker work. Previous slide. Next slide. Development of marker linked to pungency. We have already 680 SSR markers. we used to study the allelic variations among different pungent and non-pungent accessions, in which only 124 have shown polymorphic markers between, and 24 markers among 4 pungent genotypes were identified. Actually, we wanted to identify the marker for low pungent, medium pungent, and high pungent groups. So we have designed some markers, and gene-specific marker named MAPS F2 R2 targeted one locus. We have developed that marker, and it has showed polymorphism very clearly between KTPL19, which is paprika with zero pungency, against Bujolokia, habanero, and Andaman collection. And this is a partial sequencing amplified in pungent, non-pungent genotypes. The regions that we have designed this marker maps F2 bar R2, and this we could amplify in our populations. And the phenotypic variance we found in the segregating population about 67.88% in the interspecific F2 population. And we also crossed some of the Chinese accessions from Northeast and Andaman collection. We could get very good, excellent hybrids with having almost 5 kg per plant. For 3 years we have evaluated and having excellent SHU. But as I told, growing in tropical regions, now we are transferring this into capsicum annum for further, at least if we get 50% of the pungency, it will be very good. So with that objective, we are doing, work is under progress. Next, coming to the carotenoids. Carotenoids are the pigments responsible for color in hard paper. Approximately 30 pigments have been identified. The pericarp is rich source of these carotenoids. These are the different major carotenoid of contribute 60% of total carotenoids at the time of ripening. It is found, and it has an inhibitory effect, are carcinogenic compounds, which is having good medicinal value. A lot of genetic resources have been identified by various workers, and it has been published. And this is a Badgi chili with high color and low pungency, having GI tag, as we know. And normally the genetics is, it's controlled by 3 genes, and red color and other colors corresponding to Y locus gene, resulting in capsanthin capsirubin synthetase gene. And monogenic dominant nature for red fruit color, it is mentioned by Kang et al. 2000, and additive and dominance effect were found to be controlling carotenoid compounds. It is not a single gene, it is 3 genes. Again, Dr. Naresh, when in his MSc, he did this work, and we have published it, whereas non-additive gene action and additive gene action has also been reported as per genetics. These are the candidate genes which have been identified for carotenoid biosynthesis pathway. That CCS gene is responsible for red ripe color in pepper. Either presence or absence, deletion of this gene leads to the orange and yellow coloration in the peppers. This actually happened. Next, oleoresin, of course, this is oil in total which can be collected and being exported to other countries. A lot of genetic resources have been already published by many people. And
how many more?
Only 4 or 5 slides, sir. These are the genotypes with highest content with respect to biochemical compounds. Another PhD student, she has worked, and we have identified a very good genotypes which are having good level of different compounds, and significant G into E interaction we found for carotenoids and vitamins, except vitamin K. For carotenoids and vitamin synthesis, the temperature of 27, and we have worked at a different seasons. We have worked, and we found that sunshine hours of this much is very good, favorable for carotenoid production. And anthocyanin, also the cooler climate with low temperature are ideal. And genotypes which had stable and average mean performance. Here we have mentioned 2 IHR accessions which are having rich in vitamin C and total chlorophyll, and total carotenoid and vitamin E. And this anthocyanin in these 2 lines, which is highly stable with high anthocyanin content, and whereas IHR 4432, vitamin C and average stability it had. And this Badigi chili, what I was talking about, one yellow variant, what we found in the farmer's field, actually we collected it from Bagalkot farmer's field only. We found 17 BP deletion at CCS region. That is the reason the red fruit has become completely yellow, and it is very rich in lutein. We have estimated lutein. It was found very rich, and these are the overall best performing accessions having good level of biochemical parameters. And this is a natural mutant which we found. That is because deletion of 17 BP deletion of CCS gene, and it was very rich in lutein. Only one plant we found that we are just maintaining at IHR. Allelic variation where we have 17 towards 5' end coding sequence. And we found these 3 genes which are conferring for color biosynthetic genes, which we have evaluated, that CCS, on other genes were not having much effect except in IHR 4, 5, 6, 9. The yellow, orange, light red, medium red and dark red genotypes we have grouped. There is no allelic variation between different levels of color with these 3 markers, except for yellow and orange. There was some difference in the genome at genome level. So very latest study, just this month they have published, Dr. U.K. Reddy from West Virginia University. They have done wonderful work for pungency, carotenoids and flavors in chinancy, 244 accessions of chinancy through GWAS, TRAS and spatial transcriptome. This is the first integrated study which revealed key regulators of the fruit quality traits. This has given very good information which in future we can look into it. Breeders who are working. Apart from that, apart from all the health benefits, capsaicin is also being used in solar cells, which has increased efficiency of solar cells for 21.88% from 19%. It has good scope in this line also. To conclude, had peppers have very wide industrial application. There is a need to focus breeding with enhanced bioactive compounds and develop varieties or hybrids specifically suitable for these industries. Available germplasm resources can be evaluated at specified locations and can be used in breeding program. The information on candidate genes identified by various workers can be explored for identification of novel genes in existing germplasm. Allele specific markers and precise marker assisted selection will be enhancing the compounds of interest. There is need to introgress desirable genes to develop good varieties or hybrids. I would like to acknowledge my director and all the scientists who are associated, and DST, which they have funded one project on quality, and PMO CIA fellowship NUNHEMS has supported one student. He did the work here, and my both students, PhD students who did work on this quality analysis. Thank you so much.
Thank you, Dr. Madhavi. Extensive, nice.
Thank you, sir.
Thank you, and I don't think one question. Okay, you raised the hand. Quick. And then,
Madam, the pungency
Dr. Prabhu ka kidhar hai?
Huh?
pungency marker,
Huh.
marker for pungency that you have identified, right? So that marker will classify the genotype into non-pungent and pungent group, right?
Yes.
Yes.
But it will not tell the degree of pungency.
Pardon?
Degree of pungency. Like if I have 100 genotypes, this particular genotype has got 10,000 SHU or 15,000 SHU.
That we could not differentiate that using that marker.
Okay, even if the
There
marker,
is need to develop that marker actually.
okay, even if the marker identifies as non-pungent and pungent types, on what basis it will classify, madam? Like, you know, if a genotype is having 10,000 SHU, it will be classified as low pungent. If it is having more than 10,000 SHU,
With that objective only we have initiated, but we could not achieve it. So the marker what we have identified is able to differentiate between non-pungent and pungent only.
yes, madam.
There is a need to work further.
But for differentiation as non-pungent and pungent types, what is the basis, ma'am?
Here.
What is the basis?
Basis.
Can I add?
Basis.
Dr. Michael Mazurek in
What is
Cornell
that?
University. they published a paper in 2007 showing that PUN1 recessive gene in a homozygous condition gives you non-pungent material. So how it can be used today is that suppose you have a Bhut Jolokia, Chinese has excellent flavors, but you can't use it directly, only oleoresins, etc. You cross it with a non-pungent, you can cross it with a capsicum. This is my idea, how I would use it as a breeder. I'll cross it with a capsicum, and then I will use recessive PUN1 markers and get you excellent flavor capsicums in 3 back crosses. That is how practically you will use. But to get the exact Scoville units by PUN1, you will have to do a lot more work than what is already done. That's
Still,
my
we need
quick
to
answer,
work,
but it's a big,
and as
big
I
discussion.
shown the last paper, UK Reddy et al., that paper may be useful for you. That's why I mentioned this month only they published.
Thank you.
That may be used. That again in Chinese accessions, 244 accessions they have used through
Yeah.
GWAS and also transcriptome data. So that is the first of that sort. So that may be useful for us, for all
I
our
think-
breeders.
We continue the discussion.
Thank you.
It's a good discussion, but we continue. And I think now, thank you, Dr. Madhavi.
Thank you, sir.